Well, it's a pleasure to be here. I'd like to certainly thank the SPF for inviting me. I think I gave a talk at this meeting maybe 5 years ago or so, and lots happened then, so it's really great to have the opportunity to try to fill you in on some of the advances. I mean, my talk, as you can see, or my title is very broad in terms of research advances, and as you've already heard from several other speakers, you know, there's a lot of different types of advances. So I want to show mostly what we've been working on, but I also want to maybe try to put it in the context of the big picture of where I see HSP research going and a lot of the opportunities and some of the ways that we can link up with other types of researchers to encourage companies to develop therapies for the HSPs, which, as you know, are relatively rare disorders. I wanted to start out first by just a way of thinking about disease. And as many of you know, I mean, you have symptoms, the doctors you see, say you have spasticity, walking difficulties, different kinds of clinical symptoms. But I think it's important to remember, particularly in the case of genetic diseases, and even a lot of other neurologic diseases that might not be genetic, there are a lot of diseases or disturbances at the level of individual cells, which of course make us up. And this is not a new concept. I mean, as you can see, I have a quote from 1858 showing that people are really thinking about diseases as problems at the cell level. And as you can see below that, in some of the big diseases that we think about in neurology, Alzheimer's, Parkinson's, of course you know that there are symptoms associated with those that you're probably very familiar with, but not everyone's familiar with the fact that a lot of those are really diagnosed and ultimately, pathologically defined by cellular abnormalities, as in the case of plaques and tangles in Alzheimer's disease, as you can see, or Lewy bodies in the case of Parkinson's disease. So, I think that this idea of cells and targeting problems at a cell level can be very valuable for understanding and ultimately treating neurologic disease. Now, one of the really advances, I think, and I'm sure you've heard this from Corey and others over the years, that I think has really transformed HSP research is the advances in genetics. And just as an example, you can see how easy it is to sequence DNA. From the time I started at the NIH in 2001, and this only goes to 2014, but you can see that sequencing 1 million base pairs of DNA went from $8,000 to 8 cents. And there's very few examples in science of anything I've ever seen that there's been such a dramatic decrease in the cost of something, to the point where it transformed neurogenetics from a situation where you had all these people sequencing all the time to getting the DNA sequence quickly and then having to interpret it and understand what it means. And just as a comparison, you can see Moore's Law. And Moore's Law, of course, is the computing, sort of a measure of computing power. And on the left is a logarithmic scale, so it's going up tenfold each time. And you can see the dramatic change that occurred around 2008, and that was the advent of what we call next generation sequencing. And I won't go through what that means or what that entails, but it is suffice it to say that that has really transformed the ability of us to understand HSPs. And I'm going to show a little bit of an introduction. Again, I know you all know this, but I wanted to emphasize a few aspects of HSP just because it's going to be more relevant as I show some of the science. As you can see on the images on the right, we have these long neurons, these corticospinal neurons, and you heard nicely in several talks yesterday about that, that start up in your motor cortex and go all the way down to your lower part of your spinal cord. And of course, that's why the legs are affected, because those are the longest nerves. And they can be up to 1 meter in length. So they're very long cells. They're hundreds of thousands of times larger than other cell types. So they're really unique in that way. But of course, you can have dysfunction of those cells for lots of reasons, and that gives you spasticity. You can have abnormal development. You can have degeneration. You can have injury. Of course, people get spinal cord injuries. But of course, in HSPs, it's unique in that the problem is genetic. It's a specific change in different genes that gives rise to this abnormality, which again makes it different from most causes of paraplegia or paraparesis in the lower extremities. Now, as many of you know, the prevalence of HSP is somewhere between 2 and 9 out of 100,000 in different populations. But it's important to remember, too, that that's not that uncommon. That's not that much different than ALS or other diseases that are maybe more known. So I think that also emphasizes that, as people understand HSP better and better, I think that there'll be more realization and more interest in treating this disorder. Now, one of the real big advantages we have in studying HSP is the very bottom point I have there. there's about 90 or so different genetic loci. And if you think about those diseases I showed at the beginning, Alzheimer's, Parkinson's, they have far fewer genes than we do for HSPs. So we can learn a lot about HSP biology, HSP, the disease pathogenesis from all this genetic information that we have, which is really, among most diseases, is really unique. There's only a couple diseases that have this level of genetic heterogeneity that allows us to understand how these proteins might be functioning. And when I say heterogeneity, we certainly don't think of HSP as 80 or 90 different diseases. We think that these different proteins are coming together in themes that are going to allow us to sort of recategorize spastic paraplegias based on the function at the level of individual cells. And I've done this here as a sort of an example and updated it recently, but you can see that there's these different parts of the cell or different processes within a neuron that have a bunch of genes listed below them, or genes or proteins. And what those do is you can see there's certain categories that start to come out that encompass many of the different types of HSP that many of you have. And I'm just going to highlight two of them today. One is tubular ER. You can see that, and that's one of the organelles in the cell, the endoplasmic reticulum. And you can see there's lots of genes listed under there. But what's important about that is that it's the three most common forms fit into that region. So we're already thinking about half the people with HSP, could potentially benefit from any insights we have in just this area alone. And then an area of lysosomes, endosomes, and autophagosomes. And again, those are organelles within the cell, and they also have two genes, SPG11 and SPG15. I've just written the protein names there, but those are the gene names that you might be more familiar with. And those, of course, are the most common recessive forms of HSP. So if we think about those two categories, we really encompass a large number of people with HSP. And the way I tend to think about it in my own research is I tend to think of those two groups as areas where we really want to focus on. Because I also hope that if we are able to develop therapies that work in one of these, that they will cross over to some of the other types as well. Now, if you think about a cell, I showed you a neuron, and of course neurons have unique things because they have these long axons, dendrites. You heard some about that yesterday. But it's also important to remember that a lot of cells throughout the body have a lot of the same organelles. They're just maybe distributed differently. So I'm showing a picture of just a bunch of them. And this is probably something you've seen before, and people, you really learn this mostly in school. But one of the aspects of this that I think is not as clear is how crowded cells are. And I'm going to show you some images of that, how much everything is packed together. And that becomes important for how we try to understand how one organelle can affect another, or how a cytoskeletal element can affect an organelle. And I'm going to show you some new tools later on that I think are really going to transform the way we can approach, the biology of cells and how disease affects that. And I'm going to start my talk with just a couple slides about endosomal, lysosomal kind of proteins. And I just want to use this to emphasize that we already have pretty good insight into this area. And we did a little bit of work about 5 years ago on this, and several other groups have now done a lot more. But I wanted to show you that just as because it is an area I think is important, though I'm going to emphasize the ER studies for most of the rest of the talk. Now, one of the types of HSP that many of you are familiar with is SPG15 and SPG11. And I always group those together, even though they're different genes, because they make proteins that bind to each other. And if you lose one, the other one degrades. So essentially, clinically, they're the same disease. And of course, this is a disease where we know you've lost the protein. So if you're thinking about how we can treat it, well, we can treat it by trying to put the protein back there somehow through gene therapy, or we can think of ways to try to understand the biology and maybe get around this problem in some other way. Now, what's unique about these forms are that first they're recessive, or you can sometimes have parents in the U.S. and Europe and places like that in particular, where the parents have different mutations and the kids end up affected, where of course the parents don't have any symptoms at all. Now, what's different about this form of HSP with regards to the more common, what we call pure forms like SPG4 and 3A and so on, is that there's some very specific features to this. You can see in section B there's a thin corpus callosum. That's like something that is right above the ventricle on the two sort of side views of the head. And then there's sort of the other view where you see the ventricles and you see the little white things at the top of the ventricle. They call that ears of the lynx. That's a very specific MRI finding. And then it's harder to see, but in C and D, we can see abnormalities in the eye, like a different type of cataract, or we can even see some abnormalities of the retina. Now, what's valuable about these kind of things is that even if they don't affect patients, if they don't notice the symptoms, they can be very valuable for us to follow in the future for treatment, or even sometimes to be sure about a diagnosis. So these forms have other features that are involved, not just spasticity, but some of those features could be particularly valuable in the future as we develop clinical trials and we want to follow response to therapy. Now, another aspect of these that I think is very valuable is that we can see an abnormality in skin cells. So those of you that know people that have SPG 1115 or in your family know that if you look at their skin, for the most part, it looks normal. However, if we take a skin sample and we culture the cells, you can see in the Roman numeral 2-1 is a unaffected sibling, so a sister that doesn't have anything. And if you look at the lamp 1, the little thing up in the corner, you'll notice that you see lysosomal structures. And then if you look at 2 and 3, the two that are next to that, you'll notice that they're much larger. And so that's something we can see in the disease. And again, these are very, very valuable because things that are in the periphery can be very valuable for us to follow response to therapy, help with diagnosis, and so on, because they're more accessible. So again, just to summarize what we've learned about these two forms, which again are very important in this disorder, is we understand a lot about what these proteins do. We understand that they're involved in tubulation off of lysosomes. They're involved in the formation of lysosomes, and they're a type of lysosomal disorder. So as people that are working on this are trying to move toward understanding it better, oftentimes they'll come together with people studying other types of lysosomal diseases in other disorders. And again, that sort of collaboration can hopefully help us advance our understanding of these in particular. So again, I think that initial identification of what the cell biology is allows a lot more studies to occur. Now again, I'm not going to spend as much time on that. I'm going to spend most of my time back on this ER area. And again, it's partly because that's what we work on the most, but it's also important because it's the most common form of HSP. The 3 most common forms all fit into this theme. Now, if you think about the ER from your textbooks, probably when you're in school, we were always taught that it was a continuous organelle that had sheets and tubes, and they're all connected. It goes all throughout the cell. And what's remarkable when you think about that is, if you think about a neuron that's a meter long, it means your ER is a meter long. So we think about these as small organelles, but in these cells, they're not small. They really go the whole distance, and you can imagine if you disrupt the structure of that in any way, that you could run into problems with the neuron or it being able to function properly. Now, what was really remarkable, again, thinking back to the early years when we were working on this, of course, Dr. Fink discovered the first one of these proteins. It's called elastin, and that was really the way we got started in this, because I started my job around the same time that he had discovered that gene, and we were fortunate because it turned out once we started studying that, then a lot of other ones fell into place as well. And it turns out that all these proteins, as you can see on the right there, have a unique feature. They only go halfway through a membrane. And what that allows you to do is, if you think about a membrane and then you start putting things into it halfway through, what happens? It curves. And that's how you create a tube. So that's how you can have an organelle that has some part sheet, some part tubes. And a lot of these proteins, there's the most 3 common, but more and more as we go on, and there's even more than this, there's 1 or 2 more that have come out subsequently, you can already see that a lot of these spastic paraplegia genes are fitting into this same theme, to the point where if we can understand the biology of this, it's going to give us really good insights into where to go next in terms of therapies, and we're already starting to do that. Now, another thing that we don't often think about is that genes can cause many different disorders. So if you have one gene, you think, okay, I affect this gene, I'm going to get this disease. But many times we can learn a lot from what other diseases you can get. And here's an example of a study we did years ago with Michaela R. Grumbach in Austria. And what she found is that in a very large family, by segregation, which is sort of the way we used to do a lot of the identification of genes, is the same elastin protein which is, again, this ER protein that is involved in forming this ER network. A mutation that in many ways looks just like what we would see in a patient with SPG3A, instead had a completely different disease. They had a sensory disorder, a disorder of sensory nerves that gives rise to infections, and as you can see, the feet are abnormal, and some of the toes are missing because they had to be amputated because of infections. So again, this is something we don't see in HSP. So somehow this gene gave rise to a completely different disease. And you'd think that, well, we don't always understand why that happens, but I think it's very important to remember that when we think about classifying disease, neurologists were all used to classifying it by symptoms that we see. But sometimes the pathogenesis doesn't match that. And here's an example in just like a Venn diagram. We know that ALS has some features similar to HSP, though of course ALS is a much more severe disorder in terms of its progression, and there's a lot of other types of cells involved. And we know that hereditary sensory neuropathies can have overlap. As you know, HSP can sometimes have sensory abnormalities. And we sort of see this partial overlap clinically, but we also can have a common theme. So if we're thinking about trying to interest people in working on something, or pharmaceutical companies in terms of studying, or being interested in HSP, we can present to them that some of these themes that we're studying go well beyond just HSP, and we may treat diseases that normally we would think of as distinct, we may end up treating them the same way, because we're not addressing the symptoms that we see, we're addressing the underlying cause. And again, sometimes those will match up and sometimes they won't, but we can use these insights, these cellular insights, I think, to our advantage in terms of encouraging, especially companies, to pursue these, because the potential impact could be much greater than just one disease. But of course, HSP would be a beneficiary of that. Now, one of the things that's been great about these proteins that I showed you is we know a lot about what they do, and I won't go through them all, but we can essentially almost describe how you make this organelle just from genes that are affected in this disease. And in fact, a lot of our insights into this basic organelle that you'd think we would know everything about have actually come from studying the HSPs. So there's a lot of basic science interest in this as well, which again helps us tremendously because it gets more people working on these problems. Now, I know you heard in several of the talks yesterday that model systems are very important. So now we know what proteins are involved, and we also know that the ER is along the cytoskeleton. I can show, there's a picture of that there, where it moves along these. So it's not just the ER; it's how it interacts with other elements within the cell. But we really want to model this. We want to see if we start making mutations that are like people with HSP have, what does that do to a cell, and what can we do to get around that problem? And that's sort of the beginning of how we approach therapy. Now, here's an example from many years ago that we did with a group in Chicago, where we took a patient, one of the patients with HSP, took a skin biopsy, so got skin cells, and many of you have had those, and so this is what we do with those. We make those into stem cells, and then we make those stem cells into neurons. So it's like a way of making a neuron, starting with the skin. And you can see on the left, at the top is a normal cell from somebody without a disease, and you can see on the far left that there's lots of processes coming off these cells. And you'll notice at the bottom, the young girl with SPG3A, she was only 2 when we took the biopsy, you can see that the cells don't have long processes anymore. And again, that sort of makes sense. Remember, this is a disease of length, and they're shorter. But if we add certain microtubule binding drugs, again, these are drugs that we wouldn't use in patients for this because they have a lot of side effects, but it makes the point that we can reverse that. We make them go long again. And what's encouraging about that is that means we know that these are processes, these are abnormalities that can be fixed, at least in this system, and that's the beginning of therapeutics for these disorders. Another key aspect, and this is one type of model system, but remember, a neuron in a dish is not the same as an intact organism, like a human, a mouse, or a fly. So of course, you've heard about all types of models like that, and I'm going to show you a mouse model that we've made. Here's a mouse that has SPG 31, but instead of just one of the alleles missing, we took them both away. Remember, we're trying to model a disease of neurons that are this long in a mouse that's this big. So in many ways, we have to think about ways we can maybe make the mouse more severely affected, because if it was just length dependent, you might expect a normal mouse because their neurons are much smaller. So even as we heard, the circuitry inside the corticospinal neurons are similar, but they're much shorter. I mean, just because the mouse is much smaller. Now, what we can do is, if you see see this mouse on the top, you'll see that he walks just fine. You'll notice the one at the bottom who's lost this, this gene can barely walk at all. Did it work, or I wasn't sure if it actually moved? Yeah, it's a short video. I'm going to show some longer ones in a minute. But you'll notice on the right, and this is sort of something that surprised me a little bit. So these mice start getting abnormal after a couple weeks, maybe 4 weeks, 8 weeks, something like that. But if we take their spinal cords out at age 1 day, and we do recordings, and this is done with Mike O'Donovan at the NIH, we can actually see an electrophysiologic abnormality that is like spasticity, even at day 1. So we can see an abnormality of the circuitry right at the very beginning, even before we see the symptoms, which is, again, a little surprising to me, but it also emphasizes the fact that, these diseases probably start very early, and that, of course, gives us the incentive to really try to intervene as early as we possibly can with all these diseases. Now, another thing that you think about, and again, I think I'd like to emphasize, is that when I showed you those 60 genes or whatever, almost none of those are only in the brain. So it's probably not a good idea for us to think about these as only neurologic diseases. They only have symptoms that affect your nervous system in many cases, but sometimes we can use those peripheral findings, like I showed you with the skin, where we see an abnormality in the cell, but it doesn't cause any problems that you notice, but that can still be very valuable for us in terms of understanding. And I'll give you an example. Here's SPG, the same mouse. Sorry, this was right after breakfast, but this is on the left is what you see when you open the mouse up. And you'll notice on the left, there's this white stuff that's fat tissue. So we all eat a lot and you get fat tissue. But you'll notice on the right that there isn't any. And you think, well, that's a good thing, isn't it? We don't want, it's good not to have fat. But there are actually diseases that are involved in not having fat tissue called lipodystrophies. And as many of you know, if any of you have one form of HSP that is called Silver syndrome, that same gene can actually cause lipodystrophy with other different kinds of mutations. So that's another example where we have one gene giving rise to many different diseases, but this is a different gene and it's doing the same thing. So this is, again, a possible example of something that we can follow. And what we've done in D is that's a CT scan, and we can actually quantitate how much fat they have, and then we can show that they have less fat. So here, this is a good thing, maybe. So they have less fat than they would normally, and their triglycerides are lower. Now, what this emphasizes, too, is that when we think about model systems, it's important to remember models can teach us what to look back at people. So what we've done is, now that we know this mouse does this, we're doing a study with SPG 3A, 4, and 31, where we're following, some of you are in this study, following people over a period of years to see how it progresses. But one of the things we're checking is a lot of lipid profiles. Again, we're trying to see if there's some marker we can see, as we heard yesterday, how biomarkers are very important. We want to see something that's easy to access that we can follow over time to see if you respond to therapy in the future. So again, sometimes our models can give us new ideas to look back at people, and that's what we're doing in this case. And you'll notice the reason we do things like CT scans, and in the next slide, on the left there is what's called a proton MR spectroscopy, is those are non-invasive. So at the same time we're doing all these things where we're cutting sections and doing things in the mice that of course we wouldn't do in people, we're also trying to find ways that can do it non-invasively, because those are things that we're able to do to you, like MRIs and CT scans, things like that. So at the same time, we don't look at the mice just as ways to understand the biology. We're trying to develop tools that can, again, go back into studying people with the disease, and specifically the progression. Now, in this case, the proton MR spectroscopy didn't show us anything, but these are the types of things we're trying in our models. We're really trying to find tools that we can ultimately bring back up to use in patients. Now, one of the things that we've really been challenged with over the years in terms of our model systems is, and again, many of the scientists here that know that, is that they're not that robust. And drug companies would like a model that you don't have to wait for 2 years until you see a phenotype, or that it's robust enough that we can see if we make it better. And that's an important aspect of the drug discovery and development process. So one of the things we've done is we've said, well, since all these proteins work together and hitting one of them gives us a mild phenotype, what happens if we hit multiple at the same time? So here's an example of that, and I'll sort of describe it as I'm showing you this, because I haven't edited this slide to be shorter. what you'll notice is there's 4 mice, and they're on a rotor rod, so it's spinning around. The more affected ones are going to fall off fast. And you'll see the little labels on there. The one on the far right has 2 mutations. It has an elastin mutation where we've knocked in an abnormal amino acid, just like the disease, and we've taken away the REAP protein. These proteins bind to each other, and they both are involved in forming the ER. And you'll notice that the one on the right is going to fall first. The 2 next to him have just 1 mutation, and they're going to fall at about the same time. They have 1 mutation each. And the guy on the left isn't going to fall at all. So you'll notice the one on the right has fallen, and now the next 2 are going to fall at about the same time. So again, it shows you that, if we mutate 1, we get a certain kind of phenotype, and if we mutate 2, we get a much stronger one. And in fact, they have a much earlier age of onset. So again, when we start therapeutics, we'll be able to hopefully see our effects earlier, and the process will be sped up. And you'll see how much of a difference that is. That one guy fell a long time ago, and now these 2 will fall about the same time. So again, this is sort of a way to use our cellular understanding to create what I would think are more robust models. And to show you the guy on the right a little bit more, you'll see here, on the top here, this is a normal mouse, and you can see, again, walking fine. And watch how paraplegic this one looks. So you can see how just really, really dragging that hindquarters. And this is something we see at just a few months of age. So this is going to be perfect for our therapeutic trials, because also we've made this mouse so severely affected that at 6 months they're not going to live anymore. So we can also look at survival as a measure of our therapy's efficacy. Again, these are very, very prominent phenotypes, as you can see. So we've made a much more robust model that I think will be useful for us in our future studies. And I will zip through all this. What was really interesting, and again, there's not that many examples of this in neurology, and you'd think it would be more common, but the mouse actually has the cellular defect that we expect. So on the left is it's an EM image, so it's through the corticospinal tract. And you'll see on the left, that's what a normal corticospinal tract axon looks like. You see mitochondria, which are there, and there's ER going sort of longitudinally, and you can't see it because it's just difficult to see. But if you look at the mouse on the right, you'll notice something immediately. First, the mitochondria don't look normal. They're all fragmented. Those are those little sausage-like structures. But you'll also notice there's all these little lines that are going across. And that's the endoplasmic reticulum. And not only is it the ER, it's only been seen once in 20 years. There's only 1 paper ever describing a transverse form of ER. So this is something where the disease has taught us some very fundamental biology. And what's interesting is now that we know to look for this, we can see this in a fly, we can see this in a fish, we can see it in other organisms, but you never see it in culture. You only see it in an intact animal. So again, this is, when we think about cultures, remember, they're models, but they're not the same as a cell that's in an organism. And again, this is a really big finding. We have a new type of ER that we didn't know about, and this disease causes it to be stacked over and over again. So because of that, there's a lot of interest in this. And one of the things that we've been working, and this is sort of the show and tell part of my talk, and I think you'll enjoy it, we have the ability now to visualize cells at the very highest resolution that's ever been possible. Not even for HSP cells or models, but for any cell in existence. And we've been working with 3 people at Genelia, which is a Howard Hughes lab near me. And I got into this through Jennifer, who's in the middle, and she's one of the leading cell biologists in the world, and she was next to me at the NIH and then moved to Genelia. And we've been working with them to try to get better imaging, to see things better. And I think we heard about that yesterday. It's very important for us to localize things as precisely as possible. But for many years, we've had to deal with what's called confocal imaging, which has a lot of limitations. The resolution's not high enough to study many of the organelles. And Eric on the left won a Nobel Prize for this. So these machines are only available at a very, very small number of places in the world. And Harold, again, I will show you, has really pioneered a new type of electron microscopy that is far, far higher resolution than really anything that's been seen before. So just as an idea, you don't have to remember what all these acronyms mean, but there's a lot of different ways to image cells now that take advantage of what's called super resolution imaging. It overcomes, one of the big barriers of imaging is diffraction, and we have to overcome that. One of the biggest barriers of EM, electron microscopy, is that you have to cut sections. You can't slice bread infinitely narrow. So what we do is we use different technologies that get around having to slice, and we use different imaging technologies that get around diffraction. And I won't go into the physics behind it all, but I think you'll see the difference and see how much, I think, more information we're going to get from these new tools. Here's an example of the ER, and look how nice that imaging on the left looks. And that's a technique called SIM. And again, I won't go through what all these mean, but it's a super resolution type of microscopy. And you'll notice on the far right, you'll see the image at the bottom has a bunch of holes in it. And the one at the top, you don't really see that. So again, the imaging, the traditional confocal imaging, has not allowed us to see important details of the structures that we've been imaging. And here's another example of that. This is a technique called paint. What we do is, instead of imaging something through an eyepiece, you actually take single molecules and take about 1 billion of them images, and you reconstruct that, and you reconstruct a model of the cell. So what it's showing you here is this is what's called lattice light sheet microscopy, which is good but not great. And then we're going to switch to another kind of microscopy, and look at all the detail we suddenly got. So we went from imaging through our eyes to reassembling individual molecules. And again, if you do about 1 billion of those, you can get very high resolution. And we can reconstruct a cell. Again, all the membranes in a cell at a resolution that's much higher than anything you've seen. And you can see mitochondria, you can see vesicles, you can see the ER. And again, we get this really good picture of a cell. And you say, well, why do we have to get these super high resolution images? Well, as I'll show you, it's important as we understand how these HSP proteins work. And here's another example of a technique called FibSem. What that means is that, so instead of doing EM by cutting sections, maybe you can cut at 80 nanometers. We use a technique called FibSem. What it uses a focused ion beam, like people would use in making chips or something, chips like computer chips. You use this ion beam to etch a small amount of the surface off. And then you image it, and then you do it again. So it's more like polishing. You polish a little bit, take an image, polish, take an image. You get about 40-fold better resolution over cutting a section. So we get a level of detail that's not possible with EM traditionally. And I'll show you this FibSem on the left. These are all the images as we go through a cell, and you'll see what the ER looks like. We're going to reconstruct it. And then at the very bottom is what the ER would look like if you took a confocal image of it. And look at the difference. You'll see all this detail is completely lost when you take a regular confocal image. And that's actually a calculated image, so it's better than what you would see under the microscope, because we've taken all the artifacts. So look at all the detail that's completely lost. The green and the white are the same image, it's just done different ways. And this is in an area of the cell where we thought it was just two-dimensional. So we're learning that there's a lot more three-dimensionality to things, because all of our tools before just gave us 2 dimensions. So now we see much better than we ever did. And one of the things we can also see is, look at the structure at the right. We used to call that a sheet because it looked like a sheet in our traditional imaging. But you can see it's not a sheet. It's more like a honeycomb, and we call it a matrix. So it's, again, a new type of the organelle that really was never described before, but we can now see this image using these brand new tools, again, that are far better than what we've ever had before. And remember, I went back and showed you this new kind of ER. Well, we've been trying to see what that looks like. So we've been trying to actually reconstruct these 2 nanometers at a time. So I have one student who spent months just on one image, and I'll show you her work. This is an example. So if you look down through a corticospinal tract, you notice that there's all those axons. So that we're basically cutting through the tract, and what I've showed you is, as we start to reconstruct, what the organelles within that look like. So red is a mitochondria. And those are big, those are easy. ER, microtubules, and things are the other colors. But normally what you would see is just these processes going straight up. Now what we see in the model of HSP is this. You see all those ER structures are in green. The mitochondria are the other colors. It's like the yellow one and the blue one and the I guess magenta or whatever. Those are all different mitochondrial structures. And remember, this is just one little axon in a corticospinal tract of a mouse. And imagine how small that is. And look how much detail we can get using this technique. And of course, this is just a screenshot. So the resolution is much, much higher than this when we actually use the software program that we have. And the ER is separated on the left. Again, this is something we never see in a normal mouse axon. So this is an example of where the biology that we thought was going to be the case is exactly what it is. And again, that makes it a lot more promising for the future. And if we take a zoom in from the top, you can see that it has holes in it, just like that other structure I showed you. So again, I think we're starting to see a real type of endoplasmic reticulum that really is transverse in the axon. Again, no one's known about this before, and this disease has taught us it. So there's a tremendous amount of interest in this, so we will learn a lot about this in a very short period of time, and I think it will all be relevant to our understanding of this disorder. Now, I sort of wanted to end with a few kind of slides to emphasize one other aspect of biology, and that's often not shown. You know, we always look in journals and we see pictures, but it's important to remember that just as we don't examine patients or, you know, sitting perfectly still, we shouldn't examine cells sitting perfectly still. And I think people are often surprised at how dynamic cells are, and that's very important for understanding disease pathogenesis. And I'll show you an example. This is what the ER looks like with just a label. And look what happens when we look at it live. It's moving a lot. And remember, if we don't ever watch it move, we'll miss all the things that motion does. Motion could be part of the disease. And on the right, look at how fast that's moving. So one of the problems that the reason people haven't done more of this in the past is the imaging has been too slow. It's like if you take a picture with a camera and you have the shutter open for a very long period of time, what happens? Everything's blurry. All the microscopes that we use have that problem. So we can now image 200 times faster than that. So it's not even just a little bit faster, it's a lot faster. Because we can image so fast, we can see the motion a lot more clear. And that was why people didn't see those holes when they were looking at live imaging. The ER was moving so fast that it just obscured it. They never saw them. But when you can image, you know, at 100 times faster, you can see it. And here's an example on the next slide. Same thing. You know, you'll notice that the left is moving very fast. And on that left, all that imaging is on the very left image. If you took a confocal image, all that motion is happening while you're imaging. So again, you can't see it. It's a blur, and we don't have to tolerate that anymore. Which is again why these imaging tools are so valuable, I think, for the future. Now, why do we have to stop at organelles? You know, we know that your diseases are caused by individual proteins. So what if we could look at an individual protein and how it moves and how mutations affect that? And you would think that doesn't sound possible. You know, we can't possibly see that. Well, I'll show you. We can. So this is what you see on the left. All those little dots on the ER, the yellow ones, those are individual proteins moving. They're a protein that binds to one of the HSP proteins. So it's not directly one, but you can see how dynamic that is. So again, this is something we hadn't really thought about. Why is it so important that those proteins move? And does mutation affect that? And I'll show you that it does in HSP and a lot of other diseases. But again, this is something we really couldn't do. And we're imaging at 100 hertz, which again, most people image at 1 hertz. So most people would never see this motion. But I think it's going to end up being important for HSPs. And our ability to do that is going to allow us to understand how these mutations affect people. Another thing to remember, and again, sort of in the images I've showed you one organelle at a time, so I won't spend a lot of time on this, but it's also important to remember everything interacts with everything else, and we don't want to always isolate things too much. And my colleague Jennifer developed a technique a couple of years ago, and you'll see it here, where we can image all the organelles, or most of the organelles in the cell simultaneously, using 7 different color imaging. And most people do 2, and now we can do 7. And you'll see how crowded the cell is. Like, we got all the organelles moving there, and you can see they're all, they're all lit up, and the cell's pretty full. So remember, the ER is interacting with lots of things, and it's important for us to remember that as we study the biology. And again, this will go for any organelle that we're studying. It's not just the ER. I'm going to end with a couple examples of how an individual point mutation changes how a protein moves. And again, I think it's important for us to think about that as a pathogenic theme in the future. And I picked an ALS protein because it also affects the ER, and it affects where the ER contacts the mitochondria. And what I'll show you, and this is going to be hard to see, I think the people on this side will see it better because I think the blue is easier to see, but we're going to show you an individual protein and how it moves when it's normal on the top, and when we just make one little change, one amino acid change on the bottom, and it's the mutation that causes this form of ALS. And here it is, and I don't know if it's started yet. Let's see. I will start them manually, and you should see some blue moving on there. And you'll notice at the bottom there's a little circle, and the blue isn't moving at all. So what happened is when we mutate this residue, the protein stops moving, whereas in the top it moves all over the place. And again, this is an example, and it's something we don't normally think about. This is an example of how one mutation can affect an individual protein, how it moves, and we can see that. So again, we can really go all the way from the structure of a protein, to watching it move in a cell, watch how it affects the organelles, and all those insights are important in terms of understanding biology. And here's another example. I'll pick SPG4, which again is the most common form. So what happens, and I don't know if it's moving yet, I'll start it. What you'll notice at the right is one of the HSP mutations, and I don't know if it's going to show up. You notice that on the right it's not moving at all. And you'll notice on the left you see that protein moving all over the ER. It's going to go all the way around, loop around, and that's what it normally does. But if we make one mutation, just change one amino acid, it locks onto the microtubule, and you start getting these almost like aggregates form, these clusters. The microtubules are binding to the ER and they get stuck on one another. So we can actually see that at the level of just an individual protein. So look at the difference on the left and the right. So again, this is not something that's subtle, and it's just one amino acid out of 600 and something that we've changed. And it's one of the ones that causes HSP. So again, I think this is really the kind of advance that we're looking for, is that we really want to be able to understand our model systems better. And I think these tools that I showed are going to be important aspects of that. And again, if you imagine us being able to intervene, we can test our interventions at lots of different levels now that we couldn't do before. And again, I think that's going to be very valuable for the future. So just as a final slide, I think that a way to think about this is, those of us that are trained as neurologists are trained to localize lesions. But like on the textbook at the left, we go through our residencies and we learn how to do this. But you'll notice even the pictures they show, the localization is at the level of the nervous system. It's the symptoms that we see, whether it's spasticity, rigidity, all sorts of different types of things. But it's also important to localize the lesion in genetic diseases at the cell level. And it goes along with it. Both are important. The left is important as we think about symptoms and making them better. The right is important about how we can affect the process of the disease itself. And again, on the right, we know that we can link diseases that don't localize to the same part of the nervous system, but they do localize to the same part of the cell. So again, I think that when I think about the advances that we've had in HSPs, maybe in the 5 years or so since I've spoken, I think our ability to understand the biology at the individual level of what the mutations are doing has been a great advance that I think is going to continue to give us insights that allow us to develop therapies that affect the progression of the disease rather than just the symptoms that we see. And of course, the hope is that if you can affect the progression, the symptoms don't get worse. But again, it's sort of rather than a symptom-based focus, it's a mechanism-based focus of disease. And again, I'd just like to thank all of the people I work with. As I work at the NIH, and again, you can see on the right, it's the largest neuroscience building on Earth. We have about 100 neuroscientists in just one building, and we have about 60 other buildings on our campus. So it's a very, very big place. I didn't show a picture of Genalia. That's the Howard Hughes labs that allow us to do all this imaging and allow my people to spend a lot of time there. It's about 25 miles away, and they've been very generous in funding us and in supplying us with, again, equipment that isn't possible anywhere else on Earth. And again, on the left there are the people that I work with in my group. But again, I'd like to emphasize, that we work with lots of groups all over the world, and that we really benefit from that. And we're always anxious to collaborate with anyone we can to help, move this our understanding of this disease along. So thank you.
Question and one biological question.
Sure.
you saw these endoplasmic reticulum along the axon, right?
Mm-hmm.
And do you think this is also part of the problem of the microtubule, that it's like almost leakage from the cytoplasm to the axon? Or you think this neuron sends them along the axon because there is a need for, protein production or cellular function? So do you think it is like a mechanism that the cell tries to maintain homeostasis, or this is a byproduct of another ongoing problem in the cell, and that's the output that we observe them in the axon? So are they really functional, or?
Oh, I see. You're saying these transverse segments of the ER
Yes.
specifically. Okay, that's a
Yes.
good question. So, yeah, to answer that broadly, so one of the things, and I didn't show is maybe as a setup to that, is we've looked at the ER in general, like in all the processes of a neuron, and there's always ER everywhere. Even a tiny little branch, there'll be a tube going right down the middle. So the ER goes everywhere. So the distribution is very important. Now, to your point about, is this an abnormal structure? Well, we don't think so, because I can tell you when we were at Genelia, we were looking at the reconstructions that they had done of the fly brain. They had done the whole fly brain at very high resolution using FIB-SEM. They see these same structures. And one paper on this 20 years ago mentioned that it was actually something that was induced in fish by activity. So it's an activity-dependent type of ER. Now, what makes me think it's just more of a real structure is that it looks a lot like what we used to think were sheets in the periphery of every other cell. It just looks like they're oriented differently. So we don't understand the orientation. The one thing I can tell you is that in our mouse model, we see a huge increase in neurofilaments. So neurofilaments could be organizing these structures. We haven't proved that yet, but it's one possibility. So we think that they're organized through cytoskeletal interactions, but we do think it's a real type of ER that is just massively overexpressed. And again, since we didn't really even know these existed, we have to figure out what they do normally, why they're induced by activity, and then that might understand, first is, you're right, why maybe it's a result of a general dysfunction of the cell, or maybe this is the cell doing this for some particular reason, and we don't know. But that's a very important question for us to answer, and it's exactly what we want to do now, is now that we see this structure, well, what does that mean functionally? Because that gets us one step closer to understanding the disease.
Thank you so much.
Sure.
I have a question over here.
You mentioned the possibility of collaborating with other research. I happen to have, along with HSP, an autoimmune disorder called Sjogren's. It also has neurological aspects, neuropathies, that type of thing, which has made diagnosis for me very complicated. So would Sjogren's research hook in with your research?
That's a very good point. I think that's the kind of thing we need to think about more. And again, I think it's good for us to think about how this might relate to other diseases, because in some cases it's just, you can just have two different diseases, and that's just, it happens occasionally. But there's other cases where they actually might have a relationship to one another. And I think that gets back to that, I showed a lot of cell biology and things like that, but it does emphasize the importance of all the clinical studies that are done. It emphasizes that it really is important for us to really follow patients over time and work together to see whether these things really go together at a greater rate than you might see in the general population. And I think some of the people I've talked to, and we're trying one now, and several other centers is trying to improve our registries of the different forms of HSP. And again, as I started out at the beginning, now most patients are going to be able to get genetic diagnoses. The cost is going to go down of the testing. The testing is going to be better, and we're going to have a lot more opportunity to start to see some of these relationships. And again, that's one of the reasons I tried to emphasize, even with the lipodystrophy, is that sometimes it's the same gene, and sometimes it is obvious, but other times it might teach us something new. So I think that's a very important thing for us to do, is understand other symptoms that come along with different HSPs and keep an open mind on what might be related. Again, maybe at first glance we think these have nothing to do with each other, but maybe as we dig deeper we'll see that it does. And oftentimes those insights can be very important for treating either one or both of the conditions. So I think that's a very important thing for us to continue to do.
We have a question in the back.
Hi, thanks for your talk.
Sure.
You mentioned that understanding these cellular phenotypes would let you better assess the potential efficacy of certain treatments. I was wondering if you were testing any treatments in your lab, or if you had any future plans to, what kind of molecules are you
Yeah, that
gonna
is important to
explore?
do. And again, we can't always talk about this as much because a lot of us are trying to, I can tell you that companies call all the time. We come and meet with them. And what they're most interested in, I can tell you, is the models that we have. Like the cell models, like some of these that I showed you, or even the mouse models. And many times they've already developed compounds and they're already looking at HSPs. Many times, companies are set up many different ways, and some of them are set up, basically they'll be studying like a lot of cellular processes and something will just fall out. And in some cases, it appears that some of the HSPs have done that at some companies. So we're currently discussing with a lot of the companies compounds that they've already gotten through a certain level that they're interested in moving one level closer to patients, which is the preclinical level, like at the cell level and the mouse level. So that's another reason I think it's important for us to develop these, is many times the companies aren't working on this specific disease, but they have some hit that looks like it might go in that direction. For one of them, and if we have the models, it makes that process much quicker. So one of the things, certainly we do at NIH, I'm sure all the academics here, we're usually trying to foster partnerships with industry because they're better at making new compounds. This is what they do. And one of the things that we can really contribute is the expertise of understanding, what sort of effects these mutations have on different processes as we move closer to people. So again, I think that in my own experience, I would hope it's the same for the other speakers, is that the last couple of years, it means several companies have approached this. And again, it doesn't always go anywhere. Sometimes it looks like it's not going to work. But in some cases, we've met 3 or 4 times now, and I think it's going to go somewhere. So I think, again, none of this is published, so they don't even know. So these are just people that found out about it through talks. Once we publish a lot of the model systems, I think we'll get more interest. And again, we're certainly very willing to give it to anybody who wants to test things. We want to. That's why it took us 5 years to develop some of these, but the goal is to get them used, but I think that's a very important part of it is collaborating with industry, collaborating with other academics. And again, people are going to come up with compounds and ideas from lots of different ways, and many of them won't be working on HSPs, but we want to be able to make it so that it's easy for them to do that. Absolutely.
Okay, great. We've got time for one more question. One more hand. Do I see a hand? I see one right here.
Hi, Dr. Blackstone. I'm part of your current study, and just wanted recruitment possibilities. I know you're still trying to get more people. You might want to tell them,
Oh, thank
who the
you for
study's
that.
on.
Yes, I can answer the questions. Sometimes we're restricted in how we can do that, but we are doing a study now of people with SPG3A, SPG4, and SPG31, a natural history study. And one of the reasons we're doing that is, again, we're looking at some of these things that we've learned from our model systems, but we really want to see how this disease changes over time, because that's going to help us design clinical trials in the future. So that's another thing drug companies are going to look for. They want to know how these diseases change, how much of an effect is there, what measures should they use. And that's, I think, something as academics and people who see patients with these disorders that we can do already now, because then when the compounds are there, we already know how to design the trials. So I think that we're considering doing that for some of the other forms of HSP too, and certainly would encourage anyone with one of those forms. You can always go to clinicaltrials.gov. All the trials will be listed there on, in the U.S. at least. We list them there so that people can find out what's available. But certainly that's one we're doing now. And again, our hope is, one of the reasons we're doing this is we really want to sort of develop the expertise, develop the infrastructure to ultimately do the trials at the NIH. So, we certainly, that's a very, very important part of this process. I'm glad you brought that up, is we really are trying to, it seems like these are separate processes, our clinical trials versus all the stuff I showed you here, but it really is, they all fit together. And we really do want to, have that expertise and that infrastructure there so that when we do have things to test, we already really know how we want to design our trial. Again, it will make things go faster for everybody, and that's really a big goal of ours. And we want to make sure that we do the best trials we can, because we don't want a possible successful compound to not make it through because we didn't design the trial well. So we're really trying to learn that as early as we can.
Okay, everybody, give it up for Dr. Blackstone.
Thanks.